AS Level Biology 9700, Paper 3, Variant 3, October November 2025. And we are going to discuss question number 1. And question number 1 is dialysis tubing is a partially permeable membrane. Glucose molecules can diffuse through the dialysis tubing. You are required to investigate the diffusion of glucose across dialysis tubing. You are provided with the materials shown in table 1.1. 20% glucose solution is labeled with G. Distilled water is labeled with W. Benedict solution is Benedict's, and length of dialysis tubing in distilled water is labeled with D. And in this investigation, you will need to put glucose solution into dialysis tubing surrounded by water. Take samples of the water surrounding the dialysis tubing and test for the percentage or the presence of glucose in each sample of water. So you have to do these three. Now, the different steps which you have to carry out during this investigation. Carry out step 1 to step 11. Step 1 is step 1, draw a mark 8 centimeter from the top of the large test tube as shown in figure 1.1. You can clearly see this mark, which is 8 centimeter uh, away from the bottom. So step 2, remove the dialysis tubing from beaker D. Take a knot in the dialysis tubing as close as possible to one end so that the end is sealed. The whole length of the dialysis tubing needs to be separated to allow the tubing to be filled with solution. To do this, rub the whole length gently between two fingers and thumb. So this is the way the dialysis tube can only be opened. So keep it into the water and then rub it. So put 10 cubic centimeter of 20% glucose solution G into the open end of the dialysis tubing. So you have to put this glucose solution into this dialysis tubing and then rinse the outside of the dialysis tubing by dipping it in the water in beaker T, because the diameter is very small. So that is the reason the solution can be outside the dialysis tubing. So it should be rinsed. So put the dialysis tubing containing G into the large test tube and keep it in position using an elastic band. As shown in figure 1.2, you can see the knot and you can see this elastic band and you can see this large test tube and the dialysis tubing inside. So you have to put the 10 cubic centimeter of the glucose solution, and outside the tubing is the water, distilled water. Now step 7 था, what distilled water into the large test tube so that the top of the water is above the level of the glucose solution in the dialysis tubing. So distilled water should be above that level. Now you have to start the timing in step 8. You are required to take a sample of the water surrounding the dialysis tubing every 5 minutes for 15 minutes. Every 5 to 15 minutes. You will collect 3 samples. One after 5 minutes, second after 10 minutes, and the third one after 15 minutes. So in this way, you are going to select the 3 samples. Now label 3 test tubes with the times of the sample water labeled 5 minutes, second with after 10 minutes, and the third after 15 minutes. So you will label all these. So after 5 minutes, step 8, put a 1 cubic centimeter syringe into the water surrounding the how to collect this. Actually, the step 10 will show you how to collect that solution. So for this purpose, after 5 minutes, the first for this test tube, you have to put a 1 cubic centimeter syringe into the water surrounding the dialysis tubing. So you can see surrounding the dialysis tubing, and so that the end of the syringe is leveled with the mix on the level with the mark on the test tube, and then level with the mark on the test tube, remove 1 cubic centimeter from the water surrounding the dialysis tubing and put this into the test tube labeled 5. Repeat the section to remove another 1 cubic centimeter from the water surrounding the dialysis tubing and put into another test tube labeled 5. The test tube labeled 5 will now contain a 2 cubic centimeter sample. Do not stop timing. So 2 cubic centimeter, and then it will be added one more time after 5 minutes, then after 10 minutes, and then after 15 minutes. So you have to prepare in this way. Now repeat test step 10 at 10 minutes and at 15 minutes using the appropriately labeled test tubes. This one, and this one. Now repeat step 10 at 10 minutes, 15 minutes appropriately. Now you need to carry out a dilution of 20% glucose solution G. To make a 1% glucose solution, you will need 20 cubic centimeter of this 1% glucose solution. So complete table 1.2 shows the 1% glucose. So you will need 20 cubic centimeter of this 1% glucose solution. So for this purpose, you will complete table 1.2 to show the volume of distilled water W you will use to make 20 cubic centimeter of 1% glucose solution. So 1% glucose solution will be 1 cubic milliliter or 1 cubic centimeter. So to make it 20 cubic centimeter, we will add about 19. We will add about 9. We will about 9 cubic centimeter of this solution. So if it is 1, it should be 9. 90. So now the next step is step 12. Prepare the 1% glucose solution as in table 1.2. You will need to carry out a serial dilution of the 1% glucose solution you have prepared in step 12 as shown in figure 1.3. You must reduce the concentration by half between each successive dilution. You will need to prepare 4 concentrations of glucose solution in addition to 1% glucose solution from step 12. So after the serial dilution is completed, you will need to have 10 cubic centimeter of each concentration available to use. According to this given measurement, now you have to prepare 4 more concentrations. That is, in 20 cubic, 1% glucose solution of the volume, 20 cubic centimeter, there will be 1 cubic centimeter of glucose solution and 19 cubic centimeter will be of the distilled water. Now we will keep in mind this ratio and we will prepare the next concentrations now. So to prepare these next concentrations, we will move towards this table which has been shown. So complete figure 1.3 to show how you would prepare your serial dilution. Each beaker should have a labeled arrow to show the volume of glucose solution transferred, a labeled arrow to show the volume of distilled water W. Now we have to draw these two arrows over it, which will show the W. And now you can see that we are going to prepare, we had this solution that was 1% glucose solution in which we had taken 1 cubic centimeter of glucose solution and 19 cubic centimeter of distilled water. So in the next, which is half of this 1%, that is 0.5% glucose solution, we have added 10 cubic centimeter of this. 10 we have taken 10 cubic centimeter of glucose solution, and in this we will add W, that will be 10 cubic centimeter. Now we have this 0.5% glucose solution in which now we have to take 10 cubic centimeter of this solution into this, and again the 10 cubic centimeter of W we will add into it. Now the solution which will be ready now, it will be 0.2% glucose solution. Now from this solution again we will add half of this concentration, that is 0.125% glucose solution. We will again add W, which is 10 cubic centimeter. So now the solution is ready, which is 0.125% glucose solution. Now from this solution, we will make half of this concentration as adding the 10 cubic centimeter of this solution and mixing it with W, which is again 10 cubic centimeter. So in this way, we have made these 5 concentrations. One concentration they had already given us. Now we have made 4 more concentration, which is 0.5, 0.25, 0.125, and 0.0625. So in this way, this is concentration number 2, this is 3, this is 4, and this is 5. It means almost we have 5 concentrations with us for this investigation. Now moving towards the next page, and here we have reached to step number 13. Clearly and thoroughly read all these steps, then you will be able to conduct this experiment. Set up a boiling water bath ready for step 18 and step 22. Already they will provide you this water bath during your experiment. So step 14, prepare the concentration of glucose solution as shown in figure 1.3. Now you will prepare all these, that is to 1% of the given concentration and 10%, and then you will add the 10 cubic centimeter of water into it. You have prepared on last page the 4 more concentrations. Now you will put them into different beakers and label all those beakers that 1.0, 0.5, 0.125, and 0.0. So in this way, you will be able to label all these beakers. Now, label 5 test tubes with the concentrations as we have discussed right now. Now, I am moving this. You are required to carry out the Benedict test on the glucose solution that you have. For this purpose, we have to, for this step 16, put 2 cubic centimeter of 1 in the approximately labeled test tubes. Then you are required to carry out the Benedict test on the glucose solutions that you have prepared in step 14. 5 dose concentration 1%, 0.5%, 0.25%, that was 0.125%, and 0.0625%. So these were the 5 different concentrations. Now you have to perform the Benedict test on them. So the volume of Benedict solution which you will take at that time. Suppose you are taking 2 cubic centimeters. So Now you will add 2 cubic centimeter of that solution which you have taken, this 2 cubic centimeter. So the amount which you are taking for this test, then you will add the same amount of Benedict solution into it. Now, after this, after this, if the reducing sugar is present, then the color change will take place. That is it will change to blue to other patterns, as blue to green, green to yellow, yellow to orange, and orange to brick red. But keep in mind, you will use the same amount of the Benedict solution of the amount which you have taken, that is the solution which you will take if you are taking 3 cubic centimeter. But here in this step 16, they have clearly told you that you have to take 2 cubic centimeter. So you will add 2 cubic centimeter of Benedict solution into it. And now you will go to find out the accurate result by keeping in mind these color sequences. So in this way, you will get the 1 mark. Now I'm moving towards the next step number 17, and step number 17 is there. Put the volume of Benedict solutions stated in A in the test tube labeled 1%, shake gently to mix. Put this test tube into the boiling water bath and rack it to table 1.3. The time to be first color change. Time to first color change. If there is no color change after 120 seconds, stop timing and record the time as more than 120. Remove the test tube from the boiling water. Repeat step 16 to step 19 using 2 cubic centimeter of each of the other concentrations of glucose solution. Now complete table 1.3 to record your results. So it means you have to perform this Benedict test for all of these 5 concentrations. And all of these 5 concentrations, you have to record the time to first color change. That is, the first color change will be, that is from, it will be blue to green. So you will find out when the color is changed from blue to green, you will note down that time. So then you will write that time over here after observing at that time when you are performing this experiment in the lab during your exams. So you will write all those timings accurately. And in this way, if you are able to write the correct timings or the time to first color change for all these concentrations, then you will be able to get 1 mark. Now we are moving towards the next steps. You are required to carry out the Benedict test on the 3 samples collected from the water surrounding the dialysis tubing 5, 10, and 15. Step 21, put the volume of Benedict solution stated in A3 into the test tube labeled 5. Shake gently to mix. Put the test tubes into the boiling water and record in A5 the time to the first color change. If there is no color change, after 120 seconds, stop timing and record the time as more than 120. So you will note down all this timing. So remove the test tube from the boiling water bath. Repeat step 21 to step 23 for the other samples for 10 to 15 minutes. So record your results in appropriate tables. Now you have to form one table. sample and first color change. Time sample minutes per minute. So here will be 5, 10, and 15. And the timing, which will you note down for the first time to color change as we have uh, explained in the previous step. Now, after doing that, now we have reached up to this point, which is state one source of error when carrying out the Benedict test. So the one source of error which you can have during this experiment is the difficulty in determining the exact moment of the first color change. So there can be the fluctuations in the temperature when you are doing this. So the first state for this you can write difficulty in determining the exact moment of the first color change. So this can be the source of error. You can write any other source of error as you can find there are some fluctuations in the temperature of the water bath. So you can do that. So use your results in A4 and A5. Estimate the percentage concentrations of glucose in the samples taken at 5, 10, and 15%. Now you have to write the percentage concentration of all those results which you have taken in the previous steps. So you will write the correct percentage of the glucose solutions of these three different samples which you had taken. So you will match how you will come to know. So you will with the you can see the pattern which has been given for you, that is the blue to sequence, blue to green, green to yellow, yellow to orange, and orange to brick red. So in this way you will be able to find out the equations. Now suggest reason for the percentage concentrations of glucose estimated in A7. So the glucose, why we are writing this and what is the reason behind this? We know that the glucose molecules are very small and they are, you can say, uh, easily pass through dialysis tubing. So over time, more glucose solution, diffuses out of the tubing into the surrounding water. So the concentration gradient can lead to an increase in concentrations at the 5, 10, and 15 minutes. So uh, we have to estimate it for this. So I'm going to write this answer. Easily pass through the pores of dialysis tubing over time, more glucose diffuses out of the tubing into the surrounding water due to the concentration gradient, leading to an increase in concentration at the 5, 10, and 15 minutes. So in this way, you can write the answer. Now, part B of this investigation or experiment is a dialysis membrane similar to dialysis tubing is used in the treatment of kidney disease. During this treatment, the blood of a person with kidney disease is passed through a dialysis machine to remove unwanted waste products from the blood. The machine contains dialysate, which is solution of glucose and ions. Blood flows through the dialysis machine and is separated from the dialysate by a membrane. Some molecules diffuse from the blood into the dialysate into shown 1.5. You can see molecules diffusing into the dialysate molecules and solutions of the glucose and dialysate membrane is there. So this is the blood of the person with kidney disease. Now, scientists studied the movement of two molecules P and Q found in the blood of a person with kidney disease. They wanted to see if these were molecules would remain in the blood in the movement out of the blood cross. The scientists took samples of blood from a person using a dialysis machine every 5 minutes for 20 minutes and recorded the concentration of P and Q in these samples. So they have given this table 1.4 in which the time sample was taken from the blood 0, 5, 10, 15, and 20 minutes. A concentration of P in the blood arbitrary units were 200, 25, and 200, and on Q it was different from. So plot a line graph of the data in the table 1.4 on the grid in figure 1.5. Use a sharp pencil. For this purpose, you can see that you have to take these values and you have to draw the two for this two curves in this way. Well, independent variable is time sample in minutes, so we will take on x-axis. And dependent variable is concentration of P and Q. So the time which they have given us is, let me do that. In this way. Now, so the data which have been given us is, first of all, it starts from 0 and you can count. I will only give you the rough idea because on screen this is difficult for me to draw these curves. I will give you the idea, now you have to draw by yourself. So 5, after 10 is 5 small squares, use after 5 small squares, or you can make accordingly. You can take these squares according to your own. On other sides, we are going to use the values is are starting from 75 and it ends to 400. So we will start from the values over there. It will start 50, 100, 150, 200, 250, and 300, 350, and 400. So in this way, you can draw the two curves. First curve with P is the value at 0 is 200. The value at 0 is 200. So 0, this is the value. This is 200. So we will put a cross over here. The second value as is 5 is 205. 5 is 205. It will be about over here. This is a rough graph which I am going to draw. You have to draw with scale. Always use the scale to draw these. And second is at 10, there is value is again 200. At 10, again the value is 200. And then at 15, it is 195. 195. So it is nearly equal to this. And then at 20, again, it is 200. This is for P. Now for Q, the values are at 0 is 400. This is at 400. And then at 5, it is 280. At 5, it is 280. So it will be nearly equal to this value. And then at 150 at 10. At 10, it is 150. And then at 10, 150. And then at 15, it is 90. And at 20, the value is 75. So the first, draw them with scale from this point to this point, then this point and this point. This is for, this curve is for concentration P. Now the second will be in this way, that in this way, and then it will be joined with this value, with this, and then this. So it is, so in this way you can draw these graphs. But here it is little bit difficult for me to draw these graphs, so it will be very easy for you when you have a question paper and graph paper has been given over here. You can easily do that, but little bit difficult for me to find out over here. Now calculate the percentage decrease in the concentration of Q between 5 minutes and 20 minutes. Between 5 minutes and between 20 minutes, you have to find out the percentage decrease. For this purpose, I'm moving towards the graph again, the table again. At 5 minutes is 280 and at 20 minutes is 75. So percentage decreases from 28 to 75. Now we are moving towards the given space and we will be able to find out the own percent. So initial value at 5 is at 5 is 280. Final value at 20 is 75. So the decrease is 280 minus 75. It will be equal to 205. So formula for this is 205 minus 280 into 100. So if we calculate this value with calculator, then you can find out the percentage decrease, that is about 73.2%. Now suggest a reason for the rate of decrease in the concentration of Q and 0 to 10, and then from 10 to 20 minutes. First of all, 0 to 10, it is a very high decrease. That is from, the rate of decrease is very high. So this is because the concentration gradient between the blood and the dialysis is at its maximum. So the diffusion occurs most rapidly when the difference in concentration is greatest. So rate of decrease is very high because of the concentration difference between blood and distillate. So diffusion occurs most rapidly. Now 10 to 20, the rate of decrease slows down. Because the molecules move into the distillate, the concentration in the blood groups drops. So making the concentration gradient less steep. Because now the concentration gradient has has become has become less steep. So when the steepness is low, the rate of reaction will or the diffusion will also become low. So it was all about this question number 1. If you find difficulty, you can contact with me. But please like, share, and subscribe my channel. And thanks for watching. And you can watch more videos which I have been used for this purpose for your examination point of view. And I'm going to upload more videos on more papers of your for your exams.